Which one of the following is true about the importance of Lucy in resolving debates about human evolution? Big brains came before bipedalism. Lucy had a brain size of 1.8% of her body mass. Bipedalism came before big brains. Lucy was partially an arboreal primate. First up, question 1 asks about Lucy's importance in human evolution. The correct answer is bipedalism came before big brains. Here is why. Lucy, the famous Australopithecus afarensis skeleton, showed us that our ancestors were walking upright about 3.2 million years ago. Her pelvis and leg bones are key evidence. This was a game changer because it proved that walking on two legs evolved before our brains started getting significantly larger, challenging older ideas. So Lucy helped settle the debate: bipedalism first, then the big brain development. Hope that makes sense. Which one of the following can best explain behavior? It is the coordinated response of an organism to an internal or external stimuli, the tendency for parts of plants to grow towards light, the action or reactions of a person or animal in response to external or internal stimuli, a cell or a group of cells that receives and processes stimuli. Moving on to question 2, which one of the following can best explain behavior? The best fit here is choice C, the actions or reactions of a person or animal in response to external or internal stimuli. Thinking of behavior as how any organism, including us, responds to what's happening around it or inside it, is about those observable actions. Option A is a bit too broad, and B is specific to plants, that is phototropism, and D describes sensory receptors, which are part of behavior but not the definition of itself. So C is our most accurate and comprehensive answer. The quick withdrawal of your hand from a hot plate is an example of fixed action pattern, innate behavior, biological clock, key stimulus. Question 3 gives us a scenario. The quick withdrawal of your hand from a hot plate. What kind of behavior is this? The answer is innate behavior. This is a classic reflex. You don't have to learn to pull your hand away from something hot. It's an automatic, built-in response wired into your nervous system for protection. It's not a complex fixed action pattern, nor is it governed by a biological clock or a key stimulus in the way those terms are specifically used. It's a straightforward innate reflex. Which one of the following process describes a biological clock? Removal of one stem from hot object quickly, the growing of plants towards light, circannual migration of birds from temperate to tropical areas, kinesis and taxis of woollice and other simple animals. Alright, question 4 asks us, which one of the following process describes a biological clock? The correct answer is circannual migration of birds from temperate to tropical areas. Biological clocks are internal timers that regulate rhythmic behaviors. Bird migration, which happens early circannual, is a perfect example. Things like pulling your hand from heat are reflexes, plants growing towards light, is phototropism, and simple animal movements like kinesis are not governed by these complex internal clocks in the same way. So the seasonal migration of birds clearly points to a biological clock. Identify learned behavior from the following: dog trained to hunt, honeybee making honey, child withdraws hand from hot things, ants making nest. Question 5 wants us to identify learned behavior from the following. The answer is A. Dog trained to hunt. Learned behaviors are things we acquire through experience, practice, or training. They are not just instinctive. Training a dog to hunt involves teaching and reinforcing skills. Making honey, a child withdrawn from heat, that's a reflex. And ants building a nest are examples of innate, meaning that genetically programmed behaviors. Only the trained dog shows a clear learned behavior. Which one of the following features is unique to bacteria? Presence of permeable membrane, being unicellular, absence of nuclear membrane, having flagella for locomotion. Let's look at question 6. Which one of the following feature is unique to bacteria? The key unique feature is absence of nuclear membrane. Bacteria are prokaryotes, meaning their genetic material, the DNA, is not enclosed in a nucleus with a membrane. It's just a region called the nucleoid. Other options are not unique. Many cells have permeable membranes. Lots of organisms are unicellular, and the flagella are found in some eukaryotes too. The lack of a nuclear membrane is a defining trait of bacteria. This item is based on the diagram which shows the different shape of bacteria below. What is the correct name of the shape of bacteria represented by letters A, B, and C respectively? Rod spirochetes, sphere, rod, spirochete. Flagellated bacille, clusters, spirella. Spirochete, sphere, rod. Question 7 is about bacterial shapes. Referring to diagram with letters A, B, and C, the correct sequence of name is D, spirochete, sphere, rod. So A represents spirochetes, which are those flexible spiral-shaped bacteria. B represents spherical bacteria, technically called cocci, and C represents rod-shaped bacteria known as bacille. Remembering these basic shapes is fundamental for microbiology. It is possible to produce human insulin in bacterial strain. The insulin gene should be inserted in a plasmid, and the recombinant DNA plasmid should be inserted into a bacteria. What is the role of the bacteria in this process? The source of insulin and modification, the source of plasmid and insulin production, multiplying the recombinant DNA molecule, joining the gene of insulin with gene of plasmid. On to question 8, which deals with producing human insulin in bacteria. What is the role of the bacterium in this process? The answer is multiplying the recombinant DNA molecule. Here is the deal. Scientists insert the human insulin gene into a bacterial plasmid, creating recombinant DNA. When this plasmid is put into a bacterium, the bacterium as it grows and divides makes many copies of this plasmid. It acts like a teeny factory, mass producing the instructions or the recombinant DNA for making insulin. The bacterium is not the source of insulin itself or modifying it, nor is it joining the genes that happens before. Bacteria can cause disease in all of the following mechanisms except changing the genetic structure of host cells, entering living cells and disrupting metabolic systems, releasing toxins as they multiply, invading and growing on the tissues of organs. Question 9 asks, bacteria can cause disease in all of the following mechanisms except the exception is changing the genetic structure of host cell. While bacteria are nasty pathogens and can invade cells, disrupt metabolism, release toxins, and grow on tissues, they generally don't go around altering the host's DNA. That's more the territory of viruses, which can integrate the genetic material into ours. So bacteria cause trouble in many ways, but directly changing our genetic code is not typically one of them. Functional diseases differ from infectious diseases in that they are caused by microorganisms, occur in the brain and heart, are common in females and males, show no signs of damage on the organs. Question 10 explores how functional diseases differ from infectious diseases. The correct answer is D. Show no signs of damage on the organ. Functional diseases are tricky because there is no visible, measurable structural damage to the organ, like you would see with an infection or injury. Think of conditions like irritable bowel syndrome. The organ functions abnormally, but it might look normal. Infectious diseases are caused by microbes. Functional diseases are not limited to specific organs like the brain and heart and don't have a specific gender bias as defining rule. This item is based on the drawing of a bacteriophage given below. In the above diagram of a bacteriophage, DNA and protein coat are respectively represented by 2 and 3, 1 and 3, 1 and 2, 2 and 1. Question 11 refers to a diagram of a bacteriophage and asks to identify the DNA and protein code. The correct answer is C1 and 2. Looking at a typical bacteriophage structure, the genetic material, which is DNA in this case, is labeled as 1 and is found inside the head. The protective outer layer or the protein coat or capsid is labeled as 2 in number 2, forming the head and the chase structure. So 1 is DNA and 2 is the protein coat. The social impact of AIDS can be mainly revealed by fear of stigmatization, loss of appetite, reduced labor force, loss of body weight. Question 12 asks about the social impact of AIDS. The main one highlighted here is fear of stigmatization. This is a huge issue. The stigma and the discrimination faced by people living with HIV/AIDS can be devastating, leading to social rejection and fear, which can prevent people from seeking testing or treatment. While loss of appetite and weight loss are symptoms and a reduced labor force is an economic impact, the fear of stigma is a profound social burden. Which one of the following is a DNA virus? Herpes complex, HIV, hepatitis C, H1N1. Next up, question 13. Which one of the following is a DNA virus? The answer is herpes complex. Herpes viruses like the ones causing cold sores or genital herpes uses DNA as their genetic material. HIV, hepatitis, H1N1 influenza are examples of RNA virus. So remember, herpes for a common DNA virus. Which one of the following is a correct comparison of lytic and lysogenic cycle of virus multiplication? Lysogenic cycle releases new virus using only chronic cycle. Lytic cycle involves incorporation of viral DNA into the host DNA. Lytic cycle releases new virus only by splitting the host cell. Both cycles damage the host cell DNA following infection. Question 14 compares lytic and lysogenic cycle of virus multiplication. The correct statement is lytic cycle releases new viruses only by splitting the host cell. The lytic cycle is the fast and furious one. The virus replicates rapidly inside the host and then bursts out by lysing or splitting open the host cell, releasing many new viruses. The lysogenic cycle, or mentioned in A and B, involves the viral DNA incorporating into the host DNA and can dormant. Both cycles eventually damage the host, but D is too general. In HIV/AIDS community screening, students were asked to take part in voluntary testing. The following discussion presents the views of our grade 12 students named A, B, C, and D toward HIV testing. Student A: I should not take the testing. It's simply boring. Student B: we should take the testing since we are educated. We should be exemplary. Student C: if we get tested and got positive, what should we do? So let us not get tested. Student D: this HIV/AIDS testing is simply a business. We should not be cheated. Which student do you think is correct? Student B, student D, student A, student C. Question 15 presents a discussion about HIV testing among students and asks which student is correct. The best view is from student B, who says we should take the testing since we are educated. We should be exemplary. Voluntary HIV testing is crucial for public health. It helps with early diagnosis, treatments, preventing spread. So student B shows responsibility. Student A is a dismissal. C is fear, and this conspiracy theory are all unhelpful and misinformed viewpoints regarding an important health measure. Which events of cellular respiration takes place in the cytoplasm eukaryotic cell? Production of pyruvate from glucose, reaction of acetyl-CoA with oxaloacetate, production of ATP through chemosmosis, reaction of pyruvate with coenzyme A. Alright, question 16. Which event of cellular respiration takes place in the cytoplasm of eukaryotic cells? The answer is production of pyruvate from glucose. The process is glycolysis, and it is the very first stage where glucose is broken down into pyruvate, and it happens right in the cytoplasm. The other options like the Krebs cycle, B and D involving acetyl-CoA and pyruvate reaction with coenzyme A, and chemosmosis For ATP production, or option C, all occur inside the mitochondria. So glycolysis in the cytoplasm is the one. Suppose two moles of glucose is burned aerobically, how many moles of ATP can be produced by substrate level phosphorylation in Krebs cycle? 4, 6, 10, 8. Question 17 is a calculation. Suppose two molecules of glucose is burned aerobically. How many moles of ATP can be produced by substrate level phosphorylation in Krebs cycle? The answer is 4. Here is how. One molecule of glucose leads to two turns of the Krebs cycle. In each turn of the Krebs cycle, one ATP or GTP, which is equivalent to ATP here, and is made directly by substrate level phosphorylation. So, for one glucose, that is 2 ATP from the Krebs cycle. If we have two molecules of glucose, we will get double that, which is 4 moles of ATP via substrate level phosphorylation in the Krebs cycle. During alcoholic fermentation, carbon dioxide is released when acetaldehyde is reduced, pyruvate is decarboxylated, pyruvate is oxidized, ethanol is oxidized. Question 18 asks us, during alcoholic fermentation, carbon dioxide is released when? The correct point is pyruvate is decarboxylated. In alcoholic fermentation, the pyruvate, or which comes from glucose, first loses a carbon dioxide molecule, that is decarboxylation, to become acetaldehyde. And this release of carbon dioxide is what makes bread rise when yeast ferments. And then the acetaldehyde is reduced to ethanol. So the carbon dioxide release happens when pyruvate is decarboxylated. During anaerobic respiration, reduced NAD is required to split pyruvate to carbon dioxide and acetaldehyde, reduce pyruvate to lactate, converts lactate into pyruvate, reduce pyruvate to acetyl-CoA. For question 19, during anaerobic respiration, reduced NAD is required to, the answer is reduce pyruvate to lactate. In lactic acid fermentation, which is a type of anaerobic respiration, NADH, that is reduced NAD, donates its electron and hydrogen to pyruvate. This regenerates NAD+, so glycolysis can continue. And the pyruvate gets converted into lactate. This happens when your muscles are working hard without enough oxygen. Why do we get tired and fatigued during heavy exercise such as long distance running? It is due to accumulation of carbon dioxide, accumulation of lactic acid, high rate of aerobic respiration, shortage of glucose. Question 20 asks us, why do we get tired and fatigued during heavy exercise such as long distance running? The primary reason is accumulation of lactic acid. When you are exercising intensely, your muscles might not get enough oxygen for aerobic respiration. So they switch to anaerobic respiration, producing lactic acid. The lactic acid builds up, lowers the pH in your muscles, and contributes to that feeling of fatigue and burning. While carbon dioxide accumulation or glucose shortage can play roles in exhaustion generally, lactic acid is key for the specific type of fatigue. If one base in the 6 triplets of the gene coding for one of the 4 polypeptides in the hemoglobin molecule alters the triplet from guanine, adenine, guanine to guanine, thymine, guanine, which one of the following biological terms best describes the above phenomenon? Mutation, cloning, translation, transcription. Question 21 describes a change in a DNA triplet from guanine, adenine, guanine to guanine, thymine, guanine in hemoglobin gene. Which biological term best describes this? The answer is mutation. Mutation is any change in the DNA sequence. This is specific change, meaning that from guanine, adenine, guanine to guanine, thymine, guanine is a classic example of a point mutation that causes sickle cell anemia. Cloning, or option B, is making copies, whereas translation, or option C, is making protein from messenger RNA, and transcription, or option D, is making messenger RNA from DNA, and the change in the DNA itself is a mutation. Which one of the following is the basic characteristics of mutation? It is spontaneous, planned, regular, expected. Question 22 asks us which one of the following is the basic characteristics of mutation. The most fitting answer is spontaneous. Mutations are fundamentally random, spontaneous events. They can happen due to errors during DNA replication or exposure to mutagens, but they are not planned, regular, or necessarily expected in a predictive way. Their spontaneous nature is a key aspect of how genetic variation arises. The theory that suggests life can evolve autonomously from non-living objects is known as Cosmosian theory, spontaneous generation theory, biochemical theory, eternity theory. Question 23 asks about the theory that life can evolve autonomously from non-living objects. The correct term for this is biochemical theory. This theory, also known as abiogenesis, proposes that life arose from inorganic molecules through a series of chemical reactions on early Earth. The Miller-Urey experiment proved some support for this, and Cosmosian theory is about life from space. Spontaneous generation was an old disproven idea of life popping up fully formed, and eternity theory is not a scientific theory of origin. Which one of the following is true about Neo-Darwinian theory? There is a natural variation in features and variation which are heritable. All species tend to produce more offspring than possible survival. It takes into account genetics and animal behavior to understand evolution. Development of new feature is important for survival. Moving to question 24, which one of the following is true about Neo-Darwinian theory? The best statement here is there is a natural variation in features and variations which are heritable. Neo-Darwinism is essentially Darwin's theory of natural selection combined with modern genetics. It emphasizes that evolution acts on this heritable genetic variation within a population, while B is true or overproduction of offspring, and D can be part of evolution or new features for survival. A captures the core of Neo-Darwinism by highlighting heritable variation. C is partially right about genetics, but animal behavior is not its central tenet. Which one of the following statements describes the evolutionary theory of Lamarck? Acquired characteristics cannot be inherited, structures commonly used become more developed, origins of species happens only through natural selection, species start complex and gradually become simple. And our final question, 25, asks us, which one of the following statements describes the evolutionary theory of Lamarck? The correct answer is structures commonly used become more developed. Lamarck's theory, often called the inheritance of acquired characteristics, proposed that organisms could develop traits during their lifetime through use or disuse. So a giraffe stretching its neck would make it longer. He also thought these acquired traits could be passed to offspring, or which A incorrectly says they cannot be, according to Lamarck. Natural selection is Darwin idea, and D is not part of Lamarck's theory. The behavior which trainers look, if modified, could lead to the desired behavior, and reward such a behavior is innate, instinct, reflex, operant. Let's look at question 26. When trainers modify behavior to achieve a desired outcome and then reward it, what kind of behavior are they working with? The correct answer is D. Operant. Operant behavior is all about learning through consequences. Think of a dog learning a new trick for a treat. Innate behaviors, instincts, and reflexes, on the other hand, are more automatic and not primarily shaped by this kind of reward-based training. Which one of the following examples of behavior describes when an animal stops responding to stimulus or cue after a period of repeated exposures? Imprinting, reflex, habituation, conditioning. Moving on to question 27, if an animal stops responding to a stimulus after repeated exposure because it learns the stimulus is not important, what is this called? The answer is C, habituation. It's like city pigeons eventually ignoring the constant street noise because it has no real consequence for them. Imprinting, reflex, and conditioning describe different learning or response mechanisms. A policeman trained a dog on how to find explosive materials through sniffing. On another instance, the same policeman observed a week-old puppy following him on the roadside. Which one of the following is correct about the behaviors of the animals in the observation? The first case is genetically encoded, and the second is adaptive. Both of the cases indicate natural behavior of the animals. Both of the cases indicate learned behavior of the animals. The first case developed through trial, and the second genetically coded. Question 28 presents two scenarios: a dog trained to find explosive and a young puppy following a policeman. What describes these behaviors? The correct choice is D. The explosive sniffing dog learned its skills through trial and reinforcement. That is a learner behavior. The puppy following, however, is likely an example of imprinting, which is genetically coded and develops without such extensive trial-based training. Which one of the following behaviors is a courtship behavior? Touch, scent marking, ritual fighting, defending a day. Alright, question 29 asks us about courtship behavior. Which of the options fits this description for attracting a mate? The answer is C, ritual fighting. In many species, males engage in these displays or symbolic fights to demonstrate their strengths and embrace potential mates. Touch can be social, scent marking is often for treachery, and defending a dean is a protective. The Ethiopian wolf can cement its leaves and hunt in packs. During patrolling their habitat, both male and female wolves of the group maintain their area with urine-containing pheromone. This pattern of behavior best exemplifies courtship behavior, territorial behavior, operant behavior, learner behavior. Question 13 describes Ethiopian wolves using urine with pheromones to mark their habitat. What kind of behavior is this? This is a classic example of territorial behavior. They are essentially signaling ownership and boundaries to other wolf packs. Is not primarily for attracting mates or courtship, or a behavior learned through reinforcement like operant behavior. The tertiary biome, characterized by cold temperature, dry precipitation, frozen soil, and inhabited by migrating birds, is boreal forests, deserts, tropical mountain forests, tundra. Let's tackle question 31. We are looking for a terrestrial biome characterized by cold temperatures, dry precipitation, frozen soil, and inhabited by migrating birds. Which one is it? The correct answer is D. Tundra. Think of vast chilly plains with permafrost. Boreal forests have trees, deserts are dry but typically hot, and tropical mountain forests are warmer and don't feature frozen soil. This item is based on the table below. Select the correct representative for L, M, N, and O respectively. Fertile, many animals. Poor, few animals. Poor, sparse, fertile, many animals. Fertile, migratory animals. Poor, drought and fire adapting animals. Medium, ants and spiders. Poor, polar bears. Question 32 refers to a table comparing different ecosystems. We need to select the correct representative for L, M, N, and O. The answer is B. Poor, sparse, fertile, many animals. This suggests L, likely representing desert soil, is poor in nutrients, and M, desert animals are sparse. In contrast, N, such as tropical mountain forest soil, is fertile, and O, its animal life includes many animals, reflecting richer resources. Which one of the following explains biodiversity? The variability among living organisms within a species and of ecosystem, the large number of human races in the world, the similarity and difference of organisms which live in different ecosystems, larger number of organisms in the same ecosystem. What exactly is biodiversity? Question 33 asks us to define it. The most comprehensive answer is choice A, the variability among living organisms within species and of ecosystems. It's about the incredible variety of life at all levels, from genes to species to entire ecological communities, which is crucial for ecosystem health. This item is based on the table below, which shows uninvented ecosystems. In which area of the above table does all of the species are more or less equally successful? Area 2, Area 1 and 3, Area 1 and 2. Question 34 refers to a table with invented ecosystems and asks in which area all species are more or less equally successful. The correct choice is area 2. This indicates that in area 2 of the table, the population numbers for different species are relatively evenly distributed, suggesting a balanced coexistence, unlike other areas where some species might dominate. A major factor in controlling the population size that helps to minimize predators and recover prey number is disease-causing organism, predation, interspecific competition, intraspecific competition. For question 35, one major factor in controlling population size helps to minimize predators and allow prey numbers to recover. The answer is B. Predation. The dynamic interaction between predator and prey population is crucial for ecological balance, helping to regulate the numbers of both and maintaining ecosystem stability. What is the major difference that viruses have with free-living cells? Viruses reproduce only sexually, lack genetic material of their own, do not function outside of a living cell, lack locomotory structure. Question 36 asks for the major difference between viruses and free-living cells. The key distinction is C. Viruses do not function outside of a living cell. They are obligate intracellular parasites, meaning they absolutely need a host cell's machinery to replicate and carry out their life cycle. They cannot survive or reproduce independently. Why is nutrient recycling important in an environment? Because the amount of nutrients in the environment is limited. It adds new types of molecules into the environment. Nutrients in an environment will be depleted if recycled. Nutrients should be exchanged between the Earth and the other planets. Why is nutrient recycling so important in an environment? Question 37 points to the answer because the amount of nutrients in the environment is limited. Earth has a finite supply of essential elements, so these nutrients must be continuously recycled through natural processes like decomposition to support ongoing life. Which one of the following orders of life process correctly illustrates occurrence of the nutrient cycle? Feeding to death and decomposition, then excretion to respiration and breathing. Feeding to excretion to respiration and breathing, then to death and decomposition. Death and decomposition to feeding to excretion to respiration and breathing. respiration and breathing to feeding, feeding to excretion, excretion to death and decomposition. Question 38 asks for the order of life process illustrating the nutrient cycle. The correct answer is feeding to death and decomposition, then to excretion, excretion to respiration and breathing. This sequence shows organism obtaining nutrients by feeding, then nutrients returning to the environment via death and decomposition, and further cycling through excretion and the gas exchanged in respiration. Which of the following process in the phosphate cycle does not return phosphate ions to the soil? Weathering of rocks, uses of fertilizers, decomposition of dead animals, cellular respiration. In question 39, we are looking at the phosphate cycle. Which process does not return phosphate ions to the soil? The answer is D. Cellular respiration. While vital for energy production, cellular respiration primarily involves carbon compounds and energy release, not the direct return of phosphate to the soil. Weathering rocks, fertilizers, and decomposition all contribute phosphates. Bare land was gradually occupied by lichens and mosses, annual herbs, perennial herbs, shrubs, and finally by forests. Which one of the following best describes the process indicated above? Afforestation, succession, climax community, natural selection. Question 40 describes a barren gradually being occupied by different plant communities, from lichens to forests. What is the process called? This is succession, specifically ecological succession. It's natural, often predictable, step-by-step change in an ecological community over time until a stable climax community is formed. This item is based on the table below. The population number of X, Y, and Z respectively will decrease, increase, remain the same. Increase, decrease, remain the same. Decrease remains the same, increase. Remain the same, increase, decrease. Question 41 likely refers to a table showing population data for X, Y, and Z based on natality and mortality. What will happen to their numbers? The correct answer is B. X will increase, Y will decrease. Y will decrease and Z will remain the same. This is because X's birth rate exceeds its death rate, and Y's death rate is higher than its birth rate, and for Z, birth and death rates are balanced. This item is based on the table below, which shows population growth trend. Which one of the following is correct about X and Y? X is exponential growth because the numbers increase by the same fixed amount in each time period, and Y is arithmetic growth because the population doubles in each period. X and Y cannot be compared because of them grow differently with incomparable proportion. X is arithmetic growth because the numbers increase by the same fixed amount in each time period, and Y is exponential growth because the population doubles in each time period. Both X and Y are exponential growth since each of them increase regularly without interruption. This item is based on the table below, which shows population growth trend. Which one of the following is correct about X and Y? X is exponential growth because the numbers increase by the same fixed amount in each time period, and Y is arithmetic growth because the population doubles in each period. X and Y cannot be compared because of them grow differently with incomparable population. X is arithmetic growth because the numbers increase by the same fixed amount in each time period, and Y is exponential growth because the population doubles in each time period. Both X and Y are exponential growth since each of them increase regularly without interruption. Drosophila melanogaster are convenient experimental animals in genetics because they have 23 pairs of chromosomes per cell, are small animals with a short life cycle, are too cheap to breed in large numbers, have chromosomes that cannot be easily detected. Why are Drosophila melanogaster or fruit flies so convenient for genetic experiments? Question 43 gives us the answer option B. They are small animals with a short life cycle. This allows scientists to study multiple generations quickly and cost effectively, making them ideal for observing inheritance patterns. Their well understood genetics also help us. Suppose a heterozygous brown-eye father is married with a blue-eye mother. What would be the proportion of homozygous children for eye color? Hint: brown is dominant over blue. 50%, 100%, 75%, 25%. Question 44 is a genetics problem. A heterozygous brown-eyed father or genotype capital B, small letter B, uh, marries a blue-eyed mother or genotype of small letter BB. What proportion of their children will be homozygous for eye color? The correct answer stated is D, 25%. This percentage refers to the children predicted to have the homozygous recessive genotype, small letter BB, which results in blue eyes based on the provided analysis. The following question is based on the figure that shows the gains in productivity from the hybrid vigor. In the above diagram, what best describes the breeding condition represented by letter X and Z, respectively. X involves crossbreeding and Z involves inbreeding. X involves inbreeding and Z involves crossbreeding. X represents natural selection and Z represents artificial selection. Both X and Z represents inbreeding. Looking at question 45, which refers to a diagram on hybrid vigor, what breeding conditions are represented by letters X and Z? The answer is option A. X involves crossbreeding and Z involves inbreeding. X showing a peak in productivity illustrates hybrid vigor from crossbreeding, while Z often showing a decline represents the effects of continued inbreeding. If organisms share very similar molecules such as DNA, then they must be closely related evolutionarily. Which evidence of evolution supports the above information? Comparative embryology, comparative anatomy, comparative physiology, comparative biochemistry. Question 46 states that if organisms share very similar molecules like DNA, they must be closely related evolutionarily. Which evidence of evolution supports this? This is the comparative biochemistry. By analyzing and comparing the DNA, RNA, and proteins of different species, scientists can uncover evidence of common ancestry and map evolutionary relationships. Which one of the following cannot be used as an evidence of evolution through paleontology? Stomach, teeth, hair, bones. Which of the following cannot typically be used as a paleontological evidence for evolution? That's question 47. The answer is A, the stomach. Paleontology studies fossils which are primarily preserved hard parts like bones and teeth, or occasionally hair in specific conditions. Soft tissues like a stomach decompose too rarely to fossilize. The process in which individuals with a particular heritable trait survive better than the other is adaptation, evolution, selective breeding, natural selection. Question 48 describes a process in which individuals with a particular heritable trait survive better than others. What is this called? This is the core concept of natural selection. It's the key mechanism driving evolution, where advantageous traits become more common in a population over generations because they enhance survival and reproduction. Which type of selection favors individuals at both extremes of a range to have advantage over this displaying the mean value? Stabilizing selection, directional selection, natural selection, disruptive selection. Which type of selection favors individuals at both extremes of a phenotypic range, potentially leading to two distinct forms? Question 49 points to disruptive selection. This occurs when intermediate values for a trait are selected against, and individuals at both ends of the spectrum have a higher fitness. Which one of the following is true about human evolution? Humans and chimpanzees both evolved from a common ancestor. Humans evolved from chimpanzees. Humans evolved from different line of evolution than great apes. Humans evolved from great apes. Finally, question 50 asks what's true about human evolution. The correct statement is humans and chimpanzees both evolved from a common ancestor. It's a key point that humans didn't evolve from modern chimpanzees. Rather, both species diverged from a shared ancestral species millions of years ago and followed their own distinct evolutionary paths. Which one of the following organic molecule is used to make hemoglobin? Glycogen, lipids, proteins, nucleic acids. Question 51 asks which organic molecule is used to make hemoglobin. The correct answer is proteins. Think of hemoglobin as a vital protein in your red blood cells responsible for carrying oxygen. It's constructed from our complex protein chains. So glycogen is for energy storage, lipids are fats, and the nucleic acids like DNA store genetic info. All have different roles. Which one of the following is the correct formula of maltose? C6H12O6, C12H24O12, C5H10O5, C12H22O11. Moving to question 52, what's the correct formula for maltose? The answer is D, C12H22O11. Maltose is a disaccharide formed when two glucose molecules, each C6H12O6, link up. So crucially, when they join, a water molecule is removed. So if you double glucose, you get C12H24O12. But subtract the water and you correctly arrive at maltose. A grade 11 student added Benedict's reagent on food sample and heated the mixture for 5 minutes. Then he observed a color change. Which one of the following is false about the result? An orange color shows that a reducing sugar is present. A yellow color shows that a reducing sugar is present. An orange color shows that a non-reducing sugar is present. A red color shows that a reducing sugar is present. Question 53 concerns a Benedict's test. Which statement about the result is false? The answer is C. An orange color shows that a non-reducing sugar is present. This is false because an orange color in Benedict's test actually indicates a high concentration of a reducing sugar. Non-reducing sugars wouldn't give the positive result unless they are broken down first. In a Benedict test for sucrose, why does the test sample first boil with hydrochloric acid? To reduce Benedict solution, to neutralize the solution, to hydrolyze non-reducing sugar, to retest the mixture with Benedict solution. For question 54, in a Benedict's test for sucrose, why is the sample first boiled with hydrochloric acid? The correct reason is to hydrolyze non-reducing sugar. Sucrose itself is a non-reducing sugar, so it wouldn't react with Benedict's. Boiling with acid breaks sucrose down into glucose and fructose, which are reducing sugars that Benedict's reagent can then detect. Which one of the following statements explains the specificity of enzymes? Enzymes catalyze one reaction only, speed up a reaction without being used up, can be destroyed by excessive heat, can be used over and over again. Question 55 asks which statement explains the specificity of enzymes. The best choice is enzymes catalyze one reaction only. This highlights their highly specific nature, where each enzyme typically interacts with only one particular substrate or type of reaction, much like a unique key fits a specific log. Other options describe general enzyme properties but not their specificity. The relevance and the promise of biological science can be demonstrated by their contribution in finding mechanisms to feed the growing human population, finding solutions to ethnic conflicts, development of housing and construction sectors, advancement of aviation technology. Question 56 looks at the relevance of biological science. How is it demonstrated? The answer is by finding mechanisms to feed the growing human population. Biological sciences are crucial in developing higher yield crops, pest resistant plants, and sustainable farming. all vital for global food security. The other options fall more into social sciences or other technology sectors. All are the roles of biology to combat the spread of HIV/AIDS except producing drugs that can stop the virus from reproducing, breaking the transmission pathway, raising funds for infected people, producing a vaccine against the virus. Regarding question 57, all are roles of biology in combating HIV/AIDS except one. Which one? The answer is raising funds for infected people. While extremely important, fundraising is a social and economic activity. Biology's direct contribution include drug development, vaccine research, and understanding virus transmission pathways. Which one of the following statements is incorrect about AIDS? The rate of transmission of AIDS will reduce when the transmission pathway is broken. Promiscuous sexual intercourse is reduced. The number of sexual partners is restricted. Men and women are circumcised. Question 58 asks for the incorrect statement about reducing AIDS transmission. The answer is men and women are circumcised. While male circumcision does not reduce HIV transmission risk for men, the statement includes women and presents it generally. Breaking transmission pathways, reducing promiscuous intercourse, and limiting partners are all more universal, direct, and key preventive measures. Which one of the following statements explains the property of water? It takes less energy to heat water up, has the lowest latent heat of vaporization, is a transparent molecule, is highly viscous substance. Let's look at question 59 about a property of water. The correct statement is it's a transparent molecule. Water transparency is vital, allowing sunlight to penetrate aquatic environments for photosynthesis. Option A, B, and D are incorrect. Water actually has a high specific heat capacity, a high latent heat of vaporization, and a relatively low viscosity. Suppose there are two communities in a village. Community A actively participates in HIV/AIDS awareness creation, HIV prevention, and gets blood tests for HIV, whereas Community B frequently requests for food aid, participates only in afforestation activities, and HIV-positive individuals keep the secret to themselves. In which community do you think that the incidence of HIV is most probably high? In both communities, in community B, impossible to decide, in community A. Question 60 presents two communities and asks where HIV/AIDS incidence is most probably high. The answer is B, in community. This community lacks awareness efforts, does not actively participate in prevention, and high positive individuals keep their status secret. So these factors significantly increase the risk of transmission compared to community A, which is proactive. Which property of water is useful for life that it continues in a relatively warm water underneath through the cold weather? High specific heat capacity, high density in solid form than its liquid form, high surface tension, high latent heat of vaporization. Question 61 asks us which property of water helps aquatic life survive in cold weather under ice. The answer relates to option B, which concerns water's density. Critically, solid water, that is ice, is less dense than liquid water. This unique property makes ice float, forming an insulating layer that keeps the water beneath liquid and warmer, allowing life to continue. Consider the following table, which contains organic and inorganic molecules. Which rows contain the corresponding organic and inorganic molecules correctly? 2 and 3, 1 and 2, 3 and 4, 1 and 3. For question 62, we are looking at a table of organic and inorganic molecules. Which rows are correct? The answer is D1 and 3. Remember, organic molecules usually have carbon-hydrogen bonds. Row 1 correctly lists lysine and sucrose as organic and salts and hydrochloric acid as inorganic. Row 3 correctly identifies fatty acid and methane as organic and sodium hydroxide and carbon dioxide as inorganic. Cellulose is a mixture of amylose and amylopectin, a polymer of beta-glucose molecule, a non-reducing monosaccharide sugar, a polymer of alpha-glucose molecule. Question 63 asks, what is cellulose? The correct answer is a polymer of beta-glucose molecule. Cellulose is a major structural polysaccharide in plant cell walls, composed of long chains of beta-glucose units linked together. This is different from starch, which is made from alpha-glucose. Which organic molecules are formed when two fatty acid molecules are bounded to the glycerol and the place of the third is taken by a phosphate group? OXs, glycolipids, triglycerides, phospholipids. In question 64, which organic molecules are formed when two fatty acids and a phosphate group bind to glycerol? The answer is phospholipids. This structure, a glycerol backbone, two fatty acid tails, and a phosphate head, is fundamental building block of cell membranes, creating the essential lipid bilayers. All are the function of lipids, except thermal insulation, waterproofing, respiratory substrates determines a particular features. Question 65 asks, all are function of lipids except which one? The answer is option D, determines a particular features. Lipids are vital for thermal insulation, waterproofing, and as energy-rich respiratory substrates. However, determining specific inherited features is the role of genetic materials like DNA, not lipids. Three different cells are in the process of transporting materials across the same membrane as indicated below. Cell 1, water from high concentration to low concentration. Cell 2, sodium ion from high concentration to low concentration. Cell 3, glucose from low concentration to high concentration. Which type of transport mechanism do the above cells use respectively? Osmosis, facilitated diffusion, active transport. Osmosis, active transport, facilitated diffusion. Facilitated diffusion, osmosis, active transport. Facilitated diffusion, active transport, osmosis. For question 66, we have three cells transporting materials. What are their respective transport mechanisms? The correct sequence is osmosis, facilitated diffusion, active transport. Cell 1 moves water down its concentration gradient, that is osmosis. Cell 2 moves sodium ions down their gradient, likely needing a protein channel, that is facilitated diffusion. Cell 3 moves glucose against its gradient, requiring energy, thus active transport. Suppose you took a container and divided it into two halves by a membrane which is permeable to water and labeled the two halves as A and B. Finally, we added hypersonic solution in A and hypotonic solution in B. What will be the expected result of this experiment? Rise of water level in both halves, rise of the water level in B, rise of water level in A, no change in both halves. Question 67 describes an experiment with hypertonic solution in A and hypotonic solution in B, separated by a water permeable membrane. What happens? The water level will rise in A. This is due to osmosis. Water moves from hypotonic solution or higher water concentration to the hypertonic solution or lower water concentration to try and balance solid concentrations. According to the fluid mosaic model of the plasma membrane, the membrane has uniform composition of proteins. Glycoproteins form a double layer. Phospholipid bilayer forms the basic structure of the membrane. Peripheral proteins are tightly attached on the membrane. According to the fluid mosaic model of the plasma membrane or question 68, which statement is true? The answer is phospholipid bilayer forms the basic structure of the membrane. This bilayer provides a flexible and selectively permeable barrier with various proteins embedded or attached, creating the mosaic aspect. It's not a uniform protein layer. A class of grade 11 students were given two solutions A and B of unknown concentration separated by a semi-permeable membrane. The teacher instructed them to observe any changes in the water levels of the two solutions. After a few minutes, the students observed the water level in solution A has decreased. What would the students conclude from the observation? The solution B had positive water potential, solution A had higher water potential. The two solutions were isotonic, solution A was hypersonic. In question 69, students observe the water level in solution A decreases when separated from solution B. What can they conclude? The answer is solution A had a higher water potential. Water always moves osmotically from an area of high water potential to an area of low water potential. Since water left solution A, it must have started with a higher water potential than solution B. A student did an experiment and found out the catabolism of amino acid produces toxic ammonia in the liver cells, which gradually get converted into urea and excreted through urine. What would be the result of the experiment if osmosis was blocked? The liver cells gain water and become turgid, excrete more ammonia and urea, become overactive and produce more energy, become damaged and lose functionality. Question 17 considers what would happen if osmosis was blocked in liver cells processing ammonia. The most likely result is the liver cells become damaged and lose functionality. Osmosis is crucial for maintaining cellular water balance and waste transport. Without it, toxic ammonia could accumulate and cells would be unable to manage their internal environment, leading to severe damage. Consider the following key events in the study of cell biology. Hooke describes cells in a cork, Leon Hooke discovers protozoa, Sheldon and Schwann proposes a cell theory, Johnson builds the first compound microscope. Which one of the following represents the correct order from ancient to the recent? Question 71 asks for the correct chronological order of key events in cell biology. The correct sequence is B, so this means Johnson built the first compound microscope, then Hooke described cells in cork, followed by Leon Hooke discovery of protozoa, and then finally Sheldon and Schwann proposed the cell theory. One of the differences of prokaryotic and eukaryotic cells is their cell wall. This is because cell wall of prokaryotic cell is the site of energy production, impermeable, stronger than eukaryotic cells, made from sugar and amino acids. Looking at question 72, one difference between prokaryotic and eukaryotic cell walls is that the prokaryotic cell wall is, or the answer is, made to option D, made from sugar and amino acid. Specifically, many prokaryotic cell walls contain peptidoglycan, a polymer of sugars and amino acids, which provides structural support. Consider the following features of cells: have pili, have chromosome, have circular DNA, have larger ribosomes. Which of the above features of cells can be included in eukaryotic cells? 2 and 3, 1 and 2, 2 and 4, 1 and 3. Question 73 asks which features can be included in eukaryotic cells. The correct answer is option C. Eukaryotic cells possess chromosomes, or their DNA is organized this way within a nucleus, and they have larger ribosomes, typically the 18S type. Pili and a primary circular DNA genome are characteristics of prokaryotes. Which of the following is correct about the structure of cell membrane? Phospholipid molecules form a transport channel, protein molecules form a double layer, the membrane has a fluid nature that is regulated by cholesterol, proteins form the outer layer while phospholipids form the inner layer. Which statement is correct about the structure of the cell membrane? Or question 74. The answer is option C. The membrane has a fluid nature that is regulated by cholesterol. Cholesterol plays a key role in maintaining membrane fluidity, preventing it from becoming too stiff in the cold or too fluid in the heat. Proteins, not phospholipids, primarily form transport channels. Which of the following is a correct match of cellular organelles and their corresponding function? Vacuole strength and elasticity, mitochondria ATP synthesis, lysosome modification of proteins, Golgi berry digesting foreign cells. Finally, question 75 asks for a correct match of cellular organelle and its function. The correct pairing is mitochondria, ATP synthesis. Mitochondria are famously known as powerhouses of the cell because they generate most of the cell's supply of ATP, the energy currency, through cellular respiration. What structural arrangements make chlorophyll suited for its function? The floating of the photosystem and the electron transport chain in the cytosol, the fixing of the photosystem and the electron transport chain on thallic membrane, the structural differences between photosystem 1 and photosystem 2, the long distance between photosystem 1 and photosystem 2. Question 76. What structural arrangements make chlorophyll suited for its function? The correct answer is B. Think of chlorophyll as a key player in highly efficient solar power plant within the cell. Its effectiveness comes from the photosystems and electron transport chain being strategically fixed on the thallic membrane. This precise arrangement ensures that light energy captured by chlorophyll is rapidly and efficiently transferred, much like a well-organized assembly line, making photosynthesis highly effective. Why does photorespiration take place in hot environments? Because plants close their stomata and face hydrogen shortage, water shortage, carbon dioxide shortage, oxygen accumulation, oxygen shortage. Question 77. Why does photorespiration takes place in hot environment? Because plants close their stomata and face. The answer is carbon dioxide shortage. When it's hot, plants close their leaf pores or stomata to save water. Smart, right? But this also means less carbon dioxide can enter. With low carbon dioxide, the enzyme Revsco starts binding with oxygen instead, triggering photorespiration. This process actually reduces photosynthetic efficiency, all stemming from that crucial carbon dioxide shortage caused by the closed stomata. This item is based on the diagram of chloroplast given below, which letters represent the site where photolysis and carving takes place, respectively. D and F, C and E, B and A, F and E. Question 78 is based on diagram, which letters represent the site where photolysis and carving take place, respectively. The answer is C and E. In a typical chloroplast, photolysis, meaning that splitting of water using light, happens in thallic membranes, represented by C. Then the Calvin cycle, where carbon dioxide is converted into sugar, occurs in the stroma, the fluid filled space, which is E. So is C for photolysis in the thallicoids and E for the Calvin cycle in the stroma. Which of the following is correct about the process of glucose production in the light-independent reaction of photosynthesis? A total of 9 ATP molecules are synthesized. Carbon dioxide reacts with GP to form RAP. 1 TP is directly converted to 1 glucose molecule. GP is reduced to TP by reduced NADP. Question 79. Which of the following is correct about the process of glucose production in the light-independent reaction of photosynthesis? The correct choice is option D. A key step in the light-independent reaction or Calvin cycle is the conversion of GP or glycerate 3-phosphate into TP or triose phosphate. This is not just a simple switch, it's a reduction reaction. This requires energy from ATP and crucially the reducing power of NADPH thus reduce NARP to make it happen. TP is then used to build glucose. Which of the following is a correct description of the CAM pathway of photosynthesis? Carbon dioxide is stored in the form of malate during the night. The Calvin cycle takes place during the night. The Calvin cycle takes place in the bundle shell cells during the day. Carbon dioxide is stored in the bundle shell cells during the night. Question 80. Which of the following is a correct description of the CAM pathway of photosynthesis? The answer here is option A. CAM plants are masters of water conservation. They typically open their stomata at night to take in carbon dioxide. But instead of using it right away, they convert it into malate and store it. Then, during hot day, with the stomata closed to save water, they release this stored carbon dioxide from malate for photosynthesis. So, storing carbon dioxide as malate at night is key to their strategy. Which one of the following statements explain how an enzyme lowers the activation energy of a reaction? By enabling the reaction to occur at lower physiological temperature, making the individual substrate molecules more reactive, lowering the kinetic energy of the reactive molecules, changing its tertiary structure into secondary protein structure. Question 81. Which one of the following statements explain how an enzyme lowers the activation energy of reaction? The answer is option A. Enzymes are like biological catalysts. They speed up reactions. They do this by lowering the activation energy. Think of it as reducing the energy hill a reaction needs to clean. And this allows crucial biochemical reactions to proceed efficiently at normal physiological temperatures within our bodies without needing a damaging blast of extra heat. Consider the following chemical reaction below. Based on the above chemical reaction, in which class of enzymes can enzyme E be grouped? Transferase, hydrolase, isomerase, oxidoreductase. Question 82 considers the chemical reaction below. Based on the above chemical reaction, in which class of enzymes can enzyme E be grouped? The answer is D, oxidoreductase. Let's look at the reaction. RH2 loses hydrogen to become R. That's oxidation. Simultaneously, A gains hydrogen to become H2. That's a reduction. So enzymes that catalyze such oxidation reduction reactions where electrons or hydrogen atoms are transferred belong to the class called oxidoreductase. So enzyme E fits right in this group. Which one of the following enzymes is used as a washing powders? Biochemosin, lipase, cellulase, pectinase. Question 83. Which one of the following enzymes is used as washing powders? The correct answer is option B, lipase. Ever wonder how detergents tackle greasy stains? Lipase is often the hero. Lipases are enzymes that excel at breaking down fats and oils, which are lipids into smaller, more soluble parts. This makes it much easier for the water to wash away that oily green, which is why they are a common ingredient in washing powders. What happens when the substrate molecule binds to the active site or an enzyme? The substrate and the enzyme will bond permanently. They form a stable intermediate substance. The amino acid in the active site become anareactive, the chemical bonds of the substrate distorted. Question 84. What happens when the substrate molecule binds to the active site or an enzyme? The answer is option D. When a substrate locks into an enzyme's active site is not a rigid fit. The enzyme often subtly changes shape, a concept called induced fit. This interaction puts a strain on the substrate's chemical bonds, distorting them. The distortion weakens the bonds, then making the substrate more reactive and ready to be converted into products. Which of the following statements is used as a component of cofactor? Thiamine, riboflavin, ascorbic acid, retinol. Question 85. Which of the following statement is used as a component of cofactor? The correct option is B. Riboflavin. Many enzymes need helper molecules called cofactors to function. Riboflavin, also known as vitamin B2, is a vital building block of coenzymes like FAD and FMN. These coenzymes are essential partners for enzymes involved in numerous metabolic reactions, especially those crucial for energy production. So riboflavin plays a key role here. How does crossing over during meiosis bring variation in species? By increasing the amount of genes in one of the parents during meiosis, combination of genes in the gametes that are not found in either parent, combining sex and somatic cell chromosomes, creating genes that are not found in both parents due to meiosis. Question 86. How does crossing over during meiosis bring variation in species? By the answer is B. Crossing over, which happens during meiosis when sex cells form, is like a genetic reshuffle. Homologous chromosomes exchange segments of DNA. This creates new combination of genes or alleles on the chromosomes in the gametes, the combinations that weren't present in either parent's original set. This is a major way genetic variation is introduced into a species. How does our body synthesize some of the amino acids in our body? From fatty acids we consume intermediate molecules of different pathways, breaking down of pre-existing proteins, biological breakdown of our own cells. Question 87. How does our body synthesize some of the amino acids in our body? The answer is B. Intermediate molecules of different pathways. So our bodies are quite the chemists. While we get essential amino acids from food, we can synthesize non-essential ones. We do this by using intermediate molecules generated during major metabolic routes like glycolysis or the citric acid cycle. These intermediates serve as the starting points to build the specific amino acids needed. This item is based on the following list of events during cloning of an insulin gene into plasmid. The first step is obtaining insulin-producing cells and plasmid from organism, then cutting the insulin gene and the plasmid. The third one is eggs. So your job will be finding eggs. So IV or 4, inserting the recombinant plasmid into bacteria, production of insulin by bacteria, which one of the following best represents the step listed as X. Inserting the insulin gene into a bacteria, inserting the open plasmid into bacterium, fusing the insulin and plasmid genes to the cell wall of bacteria, combining the insulin gene into the open plasmid. Question 88. This item is based on the following list of events during cloning of an insulin gene into plasmid. Which one of the following best represents the step listed as X? The correct step for X is option D, engine cloning after you have obtained your insulin gene and compose it and the plasmid with restriction enzymes. X is the crucial step of ligation. So this is where the isolated insulin gene is inserted or glued into the open plasmid using an enzyme called DNA ligase. This creates the recombinant plasmid ready for the next phase. Consider the DNA molecule containing strand 1 and strand 2. Strand 1 thiamine adenine cytosine guanine thiamine adenine. Strand 2 adenine thiamine guanine cytosine adenine thiamine. What will be the sequence of the new DNA if the DNA replicates semi-conservatively? So choose the best answer. Question 89. Consider the DNA molecule containing strand 1 thiamine adenine cytosine guanine thiamine adenine and strand 2 adenine thiamine guanine cytosine adenine thiamine. What will be the sequence of the new DNA if DNA replicates semi-conservatively? The answer is D. Semi-conservative replication means each new DNA double helix has one original strand and one new strand. So if strand 1 thiamine adenine cytosine guanine thiamine adenine is a template, its new complementary strand will be adenine thiamine guanine cytosine adenine thiamine. If strand 2 or adenine thiamine guanine cytosine adenine thiamine is a template, its new partner will be thiamine adenine cytosine guanine thiamine adenine. Those two new DNA molecules will each consist of one original strand paired with its new complement, like thiamine adenine cytosine guanine thiamine adenine with adenine thiamine guanine cytosine adenine thiamine. In translation, the tRNA serves as a template of the DNA molecule. Ribosome moves forward over two codons at a time. Messenger RNA brings amino acid into the ribosomes. tRNA with complementary anticodon binds on the codon of messenger RNA. Question 19. During translation, the correct statement is option D. Translation is where the messenger RNA's genetic code is read to build a protein. tRNA molecules are the interpreters. Each RNA carries a specific amino acid and has an anticodon that's complementary to messenger RNA codon. During protein synthesis, the tRNA's anticodon binds to the matching messenger codon with the ribosome, ensuring the correct amino acid is added to the growing chain. In translation, the tRNA serves as a template of the DNA molecule. Ribosome moves forward over two codons at a time. Messenger RNA brings amino acid into the ribosomes. tRNA with complementary anticodon binds on the codon of messenger RNA. Question 91. The step of scientific method which comes after analyzing results and draw conclusion is, the answer is option D. After conducting an experiment, analyzing the data and drawing initial conclusion, the next vital step in the scientific method is to evaluate your original hypothesis based on whether your results support or contradict it. You then move to accept, reject, or modify that hypothesis for further investigation. In a group of individuals in which group A was a control and group B was an experimental group, students give a placebo to the control group and an unknown drug to the experimental group. So at the end, the experimental group got better. Which one of the following is true about the result? The drug has an effect, placebo has a better effect, the experiment was wrong, conclusion cannot be reached. Question 92. In a group of individuals in which group A was a control and group B was an experimental group, at the end the experimental group got better. Which one of the following is true about the result? The answer is option A. The drug has an effect. In this setup, the control group got a placebo and the experimental group got the drug. If the experimental group showed more improvement than could be attributed to the placebo effect alone, or which the control group helps to get, it strongly suggests the drug itself is having a positive therapeutic impact. A biologist conducted a simple experiment to investigate the effect of pH on salivary amylase. She added 5 cm3 amylase solution into 5 cm3 starch suspension with a neutral pH in a test tube. After 10 minutes, she tested it with iodine reagent and the solution turned into blue-black color. What is the dependent variable of this experiment? Applying iodine treatment, pH of the solution, adding salivary amylase, formation of blue-black color. Question 93. A biologist conducted a simple experiment to investigate the effect of pH on salivary amylase. What is the dependent variable of this experiment? The dependent variable here is option D, formation of blue-black color. In an experiment, the independent variable is what you change, or like pH, and the dependent variable is what you measure to see the effect. Here, the blue-black color with iodine indicates starch is present. If amylase works, starch disappears and the color changes. So this color, reflecting enzyme activity, depends on the conditions and is what's being observed. Which one of the following pairs of biological tools and their function is false? Petri dish is used for culturing microorganisms. Balance is used for measuring mass. Quadrat to separate solids from liquids. Optical microscope use beam of light to produce magnified image. Question 94. Which one of the following pairs of biological tools and their function is false? The false statement is C. A petri dish is for culturing microbes, balances measures mass, and optical microbes magnifies using light. Those are true. However, a quadrat is a square frame used in ecology to sample and count organisms in an area. It's not for separating solids from liquids. You would use tools like filters for that. So the quadrat's function is misstated. Grade 12 students went to a field and collected plant specimens for identification in the laboratory. They wanted to preserve the plant parts for some time and specimens can be analyzed later. Which one of the following biological tools is used for their activities? Plant press, pitfall traps, pterolites, quadrat. Question 95. Grade 12 students went to a field and collected plant specimens. Which one of the following biological tool is used for their activities? The tool they will use is option A plant press. When collecting plants for later study, you need to preserve them. A plant press does just that. It consists of a frame and layers of absorbent paper where plants are carefully arranged, then pressed and dried. This preserves the structures for identification and analysis back in the lab. Some amino acids are synthesized using transamination process in the cell. This activity more describes the cell's replication, catabolism, energy production, anabolism. Question 96. Some amino acids are synthesized using transamination process in the cell. The activity more describes the cells. The correct answer is option D, anabolism. Transamination is a process where an amino group is transferred to help form a new amino acid. And when cells are building complex molecules like amino acids from simpler components, it's called anabolism. Catabolism is the opposite, breaking things down. So the sentence is a clear example of an anabolic process. Identify the statement that correctly explains the factors of an enzyme activity. Excessive heat breaks the ionic bonds that hold the tertiary structure of an enzyme protein. Hydrogen ion concentration affects the ionic bonds of an enzyme. Increasing concentration of substrates does not affect enzyme activity. Inhibitors increase enzyme activity by increasing their turnover rate. Question 97. Identify the statement that correctly explains the factors of an enzyme activity. The correct explanation is option A. Enzymes are proteins and their specific 3D shape or tertiary structure is vital for them to work. This shape is held by bonds, including ionic bonds. Excessive heat can break these bonds, causing the enzyme to lose its shape and a process called denaturation. When an enzyme is denatured, it can no longer function effectively. Which one of the following is true about allosteric regulation? Enzymes are activated when activators are attached on the active site. Inhibition by competitive inhibitors when it interacts with the allosteric site. Activation by activators when it interacts on the allosteric site. Allosteric inhibition is occurred when inhibitors attached on the active site. Question 98. Which one of the following is true about allosteric regulation? The true statement is C. Allosteric regulation is a control mechanism for enzymes. It involves molecules binding to a site on the enzyme that is not the active site. This is the allosteric site. When an activator molecule binds to this allosteric site, it can change the enzyme shape in a way that enhances the active site's ability to bind substrate and catalyze the reaction, thereby activating the enzyme. If an enzyme from thermophilic bacteria, which catalyzes glucose 6-phosphate into fructose 6-phosphate in the process of glycolysis, is subjected to below the optimum temperature, the rate of reaction will be decreased. This is because the particles have more kinetic energy, the enzyme is denatured, more collisions between enzyme and the glucose 6-phosphate occur, less enzyme glucose 6-phosphate complex formed. Question 99. If an enzyme from thermophilic bacteria is subjected to below the optimum temperature, the rate of reaction will be decreased. This is because the reason is option D, less enzyme glucose 6-phosphate complex formed. Even for enzyme from heat-loving bacteria, there is an optimal temperature. Below this, molecules have less kinetic energy. This means fewer and less energetic collision between the enzyme and its substrate. Consequently, Fewer enzyme-substrate complex formed and directly slowing down the reaction rate. The enzyme is not denatured by cold, just less active. Cyclooxygenase 2 is an enzyme that catalyzes the conversion of arachidonic acid to prostaglandins, which promotes pain and fever in our body. The painkiller ibuprofen acts as non-competitive inhibitor of cyclooxygenase 2, reduces the production of prostaglandins. What would happen if the concentration of ibuprofen increased? Pain of the body decreased. Pain of the body is decreased. Production of prostaglandin is increased. Concentration of arachidonic acid is decreased. Substrate affinity of cyclooxygenase 2 is higher. The outcome would be A, or option A, pain on the body is decreased. Ibuprofen is a non-competitive inhibitor of COX-2, which is an enzyme that makes pain-causing prostaglandins by binding to COX-2 or not the active site. So ibuprofen reduces its activity. If you increase ibuprofen concentration, more COX-2 enzymes are inhibited, leading to even less prostaglandin production and thus a greater reduction in pain.